Okay, then we have statistics about compounds, and you may notice this is a really long page. The statistics on the frequency with which compounds appear in different metabolic reactions: 81 reactions with carbon dioxide, 68 with coenzyme A, 56 with ammonia, 53 with glutamate, 46 with oxygen, etc., etc., etc. Some protein stats: total number of polypeptides, number of them that are metabolic enzymes, number of complexes, none in this case, because in general complexes have to be curated in. Orthologs. So let's go back up here. This table identifies orthologs that are shared among all the specified organisms as well as proteins that are unique to each organism. So this ends up being a little silly because, of course, all of the enzymes are shared inside the given organism. So you're just going to have to end up scrolling through a very long list, and this is actually what makes up the bulk of this page. And so that kind of shows you which things are and are not useful to do across a single organism. Then you have a listing of multifunctional enzymes, a listing of transporters, and this is really nice. This is nice in this case. We're looking at a bug where we care a lot about the transporters. So we have uptake transporters, efflux transporters, transporters assigned to transport reactions, genes assigned to them, etc. And down below, substrate uptake, just compounds that come into the cell, compounds that come into the cell that feed into pathways, and so on. And then what's getting kicked out of the cell? Compounds that are pathway outputs, compounds that are not pathway outputs. Then how many pathway outputs are not transported out of the cell? Then a number of other things. Transcriptional information, which is generally going to be a bunch of zeros for a Tier 3 database. Genes per transcription unit, which is really interesting and can let you take a look, in this case, at the predicted transcription units. So we have 1,425 sort of directons, which are these single gene transcription units. We have 402 two-gene transcription units, etc., etc. Number of operons per pathway. I mean, it's sort of a range of statistics, probably more than you can use in one gasp. You're probably already a little bit lost and not paying attention by the time I'm down to the bottom of the page, but I'm just going to go back to the top of the page here. And the thing to remember is you can choose which one or all of these reports you want. Okay. So, at the end of the day, our comparative genomics tools let you compare different kinds of traits like reactions, pathways, compounds, proteins, and transcription across different organisms in the collection, or let you get a report on any given organism in the collection. So in part 4, we're going to look at one final type of comparison between organisms, the Ortholog Viewer. Now you saw an ortholog chart on this page, and you can look at the total set of orthologs across a number of organisms using this comparison page. But if you want to look at ortholog comparison for a given gene, there's a different way to do that, and I'll show you how to do that in part 4 of this Comparative and Omics Tools webinar series. Thank you.